A SNAB, where we make A-level biology as simple as a cell. So today we're understanding how ATP is synthesized by oxidative phosphorylation associated with the electron transport chain in mitochondria, including the role of chemiosmosis and ATP synthase. So most ATP produced during aerobic respiration is generated by oxidative phosphorylation. This process occurs on the inner mitochondrial membrane and involves reduced NAD, reduced FAD, electron transport chain, chemiosmosis, and ATP synthase. And before we go further, it's really important that you know where the location of the different stages of aerobic respiration takes place. You need to know where glycolysis takes place, you need to know where link reaction takes place, where the Krebs cycle takes place, and now where oxidative phosphorylation takes place, because these often come up as simple one-marker questions and you don't want to waste them. So the energy released from electrons moving through the electron transport chain is used to create a proton gradient, which ultimately drives ATP synthesis. Hydrogen atoms released during glycolysis, link reaction, and Krebs cycle are accepted by coenzymes. NAD accepts hydrogen atoms to form reduced NAD. FAD accepts some hydrogen atoms in the Krebs cycle to form reduced FAD. And reduced NAD and reduced FAD are important as they carry hydrogen atoms and electrons, transport them to the electron transport chain, and provide the energy needed for ATP production. Electron transport chain. The electron transport chain is located on the inner mitochondrial membrane, again location, and consists of a series of electron carrier proteins arranged in a specific sequence. The reduced NAD and the reduced FAD are oxidized, and the hydrogen atoms they carry separate into hydrogen ions and electrons. The electrons pass along a chain of electron carriers. As electrons move from carrier to carrier, they release energy in a series of small controlled steps. This prevents a sudden release, as that could possibly damage the cell, as we previously explored in an old video. The energy released by electron movement is used to actively transport hydrogen ions. The hydrogen ions are pumped from the mitochondrial matrix into the intermembrane space. This transport is against the concentration gradient of the hydrogen ions. The energy from electron transfer is converted into stored potential energy within the proton gradient. As the hydrogen ions accumulate in the intermembrane space, you should know this by now, the H+ concentration increases outside the matrix while the H+ concentration decreases inside the matrix. This creates an electrochemical gradient, and this is the difference in both ion concentration and electrical charge across a membrane. So the two components of the gradient, so the first one is the concentration gradient, so which you're probably familiar with from topic 1. So in this context, there is a higher concentration of H+ ions in the intermembrane space than in the matrix. The second type of gradient you need to be aware of is the electrical gradient. The intermembrane space becomes more positively charged than the matrix. Together, these differences form the proton motive force. So what is chemiosmosis? The movement of hydrogen ions through a membrane down an electrochemical gradient is chemiosmosis. This is an important stage, as this links the electron transport to the ATP synthesis. Hydrogen ions cannot diffuse directly through the inner mitochondrial membrane. Instead, they move through specialized protein channels. Now, the hydrogen ions diffuse from the intermembrane space into the matrix. So ATP synthase is embedded within the inner mitochondrial membrane. It forms channels through which the hydrogen ions can flow. As hydrogen ions pass through ATP synthase, the enzyme changes shape. The ADP and the inorganic phosphate bind to the active site, and the ATP synthase catalyzes the ATP formation. So again, you might be very familiar with the formula, and then ATP synthase directly produces ATP using the energy provided by the hydrogen ion flow. So now to look at oxidative phosphorylation again, this is the synthesis of ATP using energy released during electron transfer from reduced coenzymes through electron transport chain. In this process, reduced NAD and reduced FAD lose hydrogen and electrons. They are oxidized back to NAD and FAD. ADP gains the phosphate to form ATP. The mitochondrion provides the specialized structure required from ATP production. The inner mitochondrial membrane contains electron transport chain proteins and ATP synthase. The intermembrane space allows the hydrogen ions to accumulate. The matrix provides the compartment into which hydrogen ions can return to. The compartmentalization of the matrix, again, you need to really understand how the structure of the mitochondria contributes to the electron transport chain and oxidative phosphorylation, as it can come up as 3 or 4 markers. So the compartmentalization of the matrix enables a proton gradient to be established and maintained. And chemiosmosis cannot take place without this structure. The majority of ATP produced during aerobic respiration actually comes from the oxidative phosphorylation. The traditional estimate was that around 38 ATP is produced through oxidative phosphorylation. However, the modern estimate suggests that this is around 30 ATP. And the modern estimate is much more lower because some of the proton gradient is used for transport processes across the inner mitochondrial membrane rather than the ATP synthesis. That's a SNAB today. Hope you found this video useful. If you have any questions or if you need further clarifications, let me know in the comments. I'll get back to them as soon as possible. And don't forget to like, share, and subscribe.